Assalamu alaikum everyone. This video lecture is part 3 of topical past papers of photosynthesis from A2 level biology 9700. Let's start. Question number 4. Photosynthesis is an important energy transfer process. Name one chloroplast pigment. So guys, there are a lot of chloroplast pigments like chlorophyll A, chlorophyll B, beta carotene. We can write any one. So we will write chlorophyll A. We can also write chlorophyll B. We can also write beta carotene. Let's move on to part B. Explain the relationship between the absorption spectrum of the main chloroplast pigments in a species of a plant and the action spectrum for photosynthesis for that plant species. So guys, we have to explain the relationship between absorption spectrum of pigments and action spectrum for photosynthesis of the plant species. So guys, how do we define absorption spectrum? Absorption spectrum is how much of each wavelength is absorbed by a pigment. So how do we define absorption spectrum? Absorption spectrum is how much of each wavelength or color of light, how much of each wavelength or color of light is absorbed by a pigment. We can also write that absorption spectrum is the amount of absorbance of each wavelength of light by a pigment, right? And how do we define action spectrum? Action spectrum is rate of photosynthesis at each wavelength or different wavelengths at each or different wavelength of light. So guys, we all know that greater the absorption of light, greater will be the rate of reaction, right? Higher the absorbance of a particular wavelength, higher will be the rate of photosynthesis, because the light is used in the light dependent stage of photosynthesis, so there will be more photophosphorylation, more photolysis, right? Eventually producing more reduced NADP and more ATP, increasing the rate of photosynthesis. So what we can write is that higher the absorbance of light or higher the absorption of light, higher will be the rate of photosynthesis. So guys, this is the relationship between absorption spectrum and action spectrum. If absorption spectrum shows a higher absorbance of a particular wavelength of light, so at the same wavelength of light, rate of photosynthesis will be higher, because higher the light absorbance, higher will be the rate of photosynthesis, because light is used in light dependent stage, producing ATP and reduced NADP, which are used in the light independent stage or Calvin cycle to make more carbohydrates. Explain why temperature can be a limiting factor for photosynthesis. So guys, you need to know that temperature is required for optimum enzyme activity, right? So we can say that optimum temperature is required for optimum activity of enzymes working in photosynthesis, and these enzymes are such as what? Rubisco, ATP synthase, etc. So if temperature is high, or we can say increase in temperature increases the kinetic energy of substrates and enzymes. So they collide more often. Producing more enzyme substrate complexes. We all know that. How does the temperature affect the enzymes? When you increase the temperature, there are more collisions between substrates and the enzymes because the kinetic energy of enzymes and substrate increases, right? So increasing temperature increases the kinetic energy of substrates and enzymes, so they collide more often, producing more enzyme substrate complexes. And rate of light dependent stage and light independent stage increases, right? But you need to remember that if temperature is increased above optimum, what happens? If temperature is increased above optimum temperature, if temperature is increased above optimum temperature, the enzymes are denatured. And rate of photosynthesis falls. Or decreases, right? So this is a relationship between the temperature and the rate of photosynthesis, and because of this, temperature can be a limiting factor in photosynthesis. Part D, outline how the light independent stage of photosynthesis, which is actually Calvin cycle, light independent stage of photosynthesis, leads to the production of carbohydrates such as starch in plant leaves. So guys, we have to discuss Calvin cycle over here, and we have to state that how starch is made via the Calvin cycle. So if you guys remember what happens, how does the Calvin cycle starts? It starts from a 5-carbon compound called RuBP, and you all know that RuBP combines with what? It combines with carbon dioxide, right, to form a 6-carbon intermediate, which splits into what? It splits into GP, and then GP is converted to TP, right? So for the combination of carbon dioxide with RuBP, an enzyme is required, that is Rubisco. So Rubisco catalyzes the combination of carbon dioxide with RuBP to form an unstable 6-carbon intermediate, which is converted to GP. GP is a 3-carbon compound, and GP is then converted to TP. TP is also a 3-carbon compound. So for the conversion of GP to TP, ATP is required, right? So ATP is broken down into ADP and inorganic phosphate, right? What else is required for the conversion of GP to TP? Reduced NADP is also required, right? So you need to mention this. Once TP is made, you know that most of the TP is used to regenerate RuBP, but this is not our topic right now. We want to make starch from TP. So TP is used to make glucose or hexose, and then glucose is used to make starch. So we have to follow this pathway over here. And you need to remember that when glucose is used to make starch, so obviously there is a condensation reaction, glycosidic bonds are made. So we have to mention that as well, right? So let's write over here the answer of this question. We will write that RuBP combines with carbon dioxide or CO2, carbon dioxide, to form GP, right? Initially 6-carbon compound is made, but that 6-carbon compound or 6-carbon intermediate splits into 2 molecules of GP. So RuBP combines with carbon dioxide to form GP. This is carbon dioxide fixation. And is catalyzed by Rubisco, right? Then what happens is that GP is converted to TP. GP is converted to TP using ATP and reduced NADP, right? What happens next is that TP is used to make glucose, and glucose is used to make starch. Glucose is used to make starch by condensation reaction. By condensation reaction, you can also mention that glycosidic bonds are formed. So guys, that's it. We are done with this question. Now we will move on to the next question. Let's discuss question number 5, part A. Photosynthesis is an energy transfer process that takes place in chloroplasts of plant cells. Part 1, figure 3.1 shows a diagram of a chloroplast. Using the letters A, B, C, or D, identify the structure that contains genes that code for some of the enzymes used in photosynthesis. So guys, which structure of the chloroplast contains genes? It's the circular DNA, and these are the circular DNA molecules that we can see in the chloroplast. So guys, D is the answer for the first statement, because D contains the genes that code for some enzymes used in photosynthesis. Alright, what is the site of synthesis of some of the enzymes used in photosynthesis? So guys, the first statement was regarding the genes that code for the enzymes used in photosynthesis. The second one is the site of synthesis of some enzymes used in photosynthesis. So you need to know that the enzymes are proteins, and proteins are synthesized in 70S ribosomes in the chloroplast. So guys, B is the answer for this, because B is the 70S ribosomes, which are the sites of protein synthesis or the sites of enzyme synthesis in chloroplast. Let's move on to part 2. Explain how grana are adapted for their specific role in photosynthesis. So guys, if you recall grana are stacks of thylakoid, right? What are grana? Grana are the stacks of thylakoid, and we can see these are grana, right? So we need to explain how grana are adapted for their specific role in photosynthesis. So you need to know that grana are involved in the light dependent stage of photosynthesis, right? So grana are basically involved in cyclic and non-cyclic photophosphorylation. They are also involved in photolysis, which occur during non-cyclic photophosphorylation. So we simply have to answer that how grana are adapted for their specific role in photosynthesis. So guys, you need to mention that grana are the stacks of thylakoid, right? Grana are stacks of thylakoid, and because of this there is large surface area, right? Grana are stacks of thylakoid which increases the surface area, for maximum light absorption, right? As you all know that grana contains photosystems, which are light harvesting clusters that absorb light energy. So you can see that grana are basically stacks of thylakoids, and in grana there are a lot of membranes, right? Because grana are stacks of thylakoids, grana are piles of thylakoids, so there are a lot of membranes, and this increases the surface area for maximum light absorbance. All right, so grana are stacks of thylakoid which increases the surface area for maximum light absorbance or light absorption. We can also write that grana contain photosystems which contain photosynthetic pigments for absorbance of light, right? So this is another adaptation of grana for its function, right? Grana contains photosystems or grana contain photosystem which contain photosynthetic pigments for absorption of light. Thylakoid membranes in the grana. Thylakoid membrane in grana also contains what? Also contains electron transport chain, or ETC. Electron transport chain to transport electrons, to transport excited electrons, right? As we all know that electrons are excited by the absorption of energy, and electrons are released from the photosystems, right? And then these electrons are picked up by electron transport chains, and electron transport chains transport these excited electrons to release energy, right? Thylakoid membranes in the grana also contain electron transport chain to transport excited electrons to release energy, which is used to pump protons or hydrogen ions from stroma to thylakoid space, right? Setting up a proton gradient. Then you need to mention that there is also thylakoid space in grana, right? And what is the purpose of thylakoid space? There is thylakoid space which holds what? Which holds hydrogen ions, or we can say which contains hydrogen ions, or builds up hydrogen ions in order to set up what? In order to establish or set up proton gradient. So guys, thylakoid space or thylakoid spaces in the grana are also an adaptation, because these spaces can build up proton or hydrogen ions, setting up proton gradient, right? And you need to also know that thylakoid membrane is relatively impermeable. Thylakoid membrane is relatively impermeable. What? Impermeable to? Relatively impermeable to protons. This maintains what? This maintains a proton gradient. What is meant by this statement? Because thylakoid membrane is relatively impermeable to protons, so protons cannot cross the thylakoid membrane, right? Protons cannot leak through the thylakoid membrane, and because of this, protons are captured in the thylakoid space, setting up a proton gradient across the thylakoid membrane between the stroma and the thylakoid space, right? So thylakoid membrane being relatively impermeable is also an adaptation. Thylakoid membrane being relatively impermeable to protons is also an adaptation, right? Then we can say there is ATP synthase to make ATP. There is ATP synthase where? In grana or in the thylakoid membrane to make ATP. There is ATP synthase to make ATP. So guys, these all are adaptations of grana for their specific role in the photosynthesis, that is the light dependent stage. Let's move on to part B. An experiment was carried out to investigate the effect of changing light conditions on the pH of the chloroplast stroma. Scientists followed pH changes in chloroplast stroma using fluorescent chemicals that can be used as pH indicators. So guys, in this investigation, they are investigating the changes in the pH of stroma in changing light conditions. So the steps were: chloroplasts were isolated from cells. A suspension of chloroplasts was prepared and kept in dark for 180 seconds. The chloroplasts were exposed to a period of light of fixed light intensity for 240 seconds, then returned to dark conditions. The pH of the chloroplast stroma was continuously measured and recorded. Figure 3.2 shows the results of this experiment. So guys, what they have done is that they kept chloroplasts in dark initially for 180 seconds, then they placed the chloroplasts in light for 240 seconds, and then the chloroplasts were returned to dark, and we can see the changes in the pH. of what? Stroma of the chloroplast. Describe the results shown in figure 3.2. So guys, how do we describe a graph, or how do we describe the results shown in a graph? We just need to discuss the trend. We don't need to give any reason. We are not explaining the results. We just need to describe the results. So we can see over here that pH is low in dark initially, and then pH increases sharply as the chloroplasts are placed in light, right? So what we can write over here is that pH of stroma increases sharply as chloroplasts are placed in light from dark. Or we can say pH of stroma increases sharply as conditions are changed from dark to light. So pH of stroma increases sharply as chloroplast conditions are changed from dark to light. Then we can see that in the light, what is happening? There are minor fluctuations, but kind of the pH levels off in light. So we can mention that pH of stroma levels off in light. There are minor fluctuations, but we can see that the pH is kind of leveling off in the light. And then when the chloroplasts are placed in dark again, the pH falls gradually. So we can mention over here that pH of the stroma falls gradually. pH of the stroma falls gradually as the chloroplasts are placed in dark again from light. So guys, this is the description of the results. We can write more points, we can quote values as well, but this question is of 3 marks, so that's it, right? We have written 3 points. This question is we have written 3 points. This question is of 3 marks. So that's it. If you want, you can also write other points. You can write or quote values. Let's move on to part 2. Discuss how the results in figure 3.2 support that chemiosmosis occurs during photophosphorylation. So guys, photophosphorylation occurs during light. You need to know, and you already know this, that photophosphorylation occurs during light. And the results over here show that in light, pH of the stroma rises. So how does this support chemiosmosis during photophosphorylation? So guys, if we just draw quickly a mini diagram of the thylakoid membrane with thylakoid space, right? So this is the thylakoid membrane with thylakoid space. This is thylakoid space. And what is this? This is stroma, right? And this is the electron transport chain, right? All right. And this is the photo system. So when light strikes onto the photosystem during photophosphorylation, electrons are excited and they're removed from the photosystem, right? For example, this is photosystem 1. Then these electrons are picked up by electron transport chain, right? And the electrons flow along the electron transport chain. And as the electrons flow, what happens? Energy is released, right? and the energy that is released by the electron transfer is used to pump what? It's used to pump protons from the stroma to the thylakoid space, right? So what will happen? We are just discussing photophosphorylation right now. Protons are pumped from the stroma to the thylakoid space. Thylakoid space already has a higher proton concentration or a hydrogen ion concentration. So hydrogen ions are being simply pumped from the stroma to the thylakoid space. As the hydrogen ions are pumped from the stroma to the thylakoid space, obviously hydrogen ion concentration in the stroma will fall and pH will rise. And this is true from the results that we can see. In light, pH of the stroma rises. And we can see over here that during photophosphorylation, what is happening? pH of the stroma is rising. Why? Because hydrogen ions are being pumped from the stroma to the thylakoid space. Hydrogen ion concentration falls and pH rises. And obviously, when the proton gradient is set up, there is a very high concentration of protons in the thylakoid space and low concentration in the stroma. What happens? These hydrogen ions are then diffused down the concentration gradient back into the stroma through the ATP synthase, producing ATP from ADP and inorganic phosphate. This process is called chemiosmosis. What is chemiosmosis? Diffusion of hydrogen ions down the gradient from the thylakoid space to the stroma via what? ATP synthase, producing ATP. So guys, the results over here are suggesting that chemiosmosis occurs during photophosphorylation because the pH of the stroma increases. So let's write over here that light energy causes electrons to get excited and to get released from the photosystems then we can mention that electrons are picked up by electron transport chain, and we can just write it very short that electrons move along electron transport chain. Which electrons? Excited electrons move along electron transport chain, releasing energy to pump protons from stroma to the thylakoid space, This decreases what? This increases the pH of stroma as shown in figure 3.2, right? Then we can say hydrogen ions or protons diffuse back into the stroma through ATP synthase, producing ATP, This is what? Chemiosmosis. So chemiosmosis is suggested by the results in figure 3.2, right? This is chemiosmosis. So guys, we are done with this question. Thank you so much for watching. Allah Hafiz everyone.